Today, we're talking about mitochondrial respiratory chain and mitochondrial disease. Okay, if you're unfamiliar with how the podcast goes, an AI, Grog, and I talk about topics I'm unfamiliar with, and the AI explains these topics. Please take everything it says with a pinch of salt, cross reference everything you hear. However, I do hope you find the video enjoyable and informative. Let's start today's topic, Grog. Give us an introduction.
All right, let's dive into the mitochondrial respiratory chain and mitochondrial disease. The mitochondrial respiratory chain, also called the electron transport chain, is a series of protein complexes in mitochondria that generate ATP, the cell's energy currency, by transferring electrons through a process called oxidative phosphorylation. Mitochondrial diseases are dysfunctional mitochondria, often due to genetic mutations, leading to energy production issues that can affect organs like the brain, muscles, and heart. Ready to start with a specific aspect, like how the chain works or symptoms of these diseases?
Well, yeah, tell me about the electron transport chain. So it's four large multi-unit proteins in the inner mitochondrial membrane. Why specifically in the inner membrane? Why four? Why proteins?
The electron transport chain, ETC, sits in the inner mitochondrial membrane because this membrane has a large surface area due to its cristae folds, is impermeable to most molecules, and creates a proton gradient essential for ATP production. The ETC consists of four large protein complexes, 1 to I, because each handles specific electron transfer steps, using proteins since they can precisely bind and shuttle electrons and protons via specialized structures like heme groups and iron-sulfur clusters. These complexes, along with mobile carriers like ubiquinone and cytochrome C, work together to transfer electrons from NADH and FADH2 to oxygen, driving proton pumping to power ATP synthesis. Want to zoom in on the complex's roles or why the inner membrane structure is key?
So so the electron transport chain catalyzes reactions. For instance, the oxidation of NADH. And how exactly does the energy released? First of all, why is energy released? And second, how does the energy released relate to ATP production?
All right, let's break this down. The electron transport chain, ETC, catalyzes the oxidation of NADH and FADH2, where NADH donates electrons to complex 1, releasing energy because electrons move from a high energy state in NADH to a lower energy state as they pass through the complexes to oxygen, the final electron acceptor. This energy release drives complexes 1, 3, and 4 to pump protons, H plus R, across the inner mitochondrial membrane, creating a proton gradient. That gradient powers ATP synthase, a protein that uses the energy from protons flowing back into the mitochondrial matrix to convert ADP to ATP, producing the cell's energy currency. Want to dive deeper into the proton gradient or ATP synthase's mechanism?
Well, I want to talk about the components of the electron transport chain. So we have four complexes, 1, 2, 3, and 4. Wow, what a surprise. So tell me about how they're linked to soluble proteins such as ubiquinone and cytochrome C.
All right, let's dive into the components of the electron transport chain, ETC. The ETC has four main protein complexes, complex 1, 2, 3, and 4, embedded in the inner mitochondrial membrane, linked by two mobile, soluble carriers, ubiquinone, also called coenzyme Q10, and cytochrome C. Complex 1, NADH dehydrogenase, takes electrons from NADH and passes them to ubiquinone, a lipid-soluble molecule that shuttles electrons freely in the membrane, to complex 3, cytochrome BC1 complex. Complex 2, succinate dehydrogenase, feeds electrons from FADH2 into ubiquinone as well, bypassing complex 1. Complex 3 then transfers electrons to cytochrome C, a small protein that moves along the membrane's outer surface to deliver electrons to complex 4, cytochrome C oxidase, which finally hands them to oxygen, forming water.
Okay, can
These
you explain very simply what these things do and why we need them?
four complexes of the electron transport chain, I, 2, 3, and 4, take electrons from molecules like NADH and FADH2 and pass them along, using carriers like ubiquinone and cytochrome C to oxygen, which makes water. This process releases energy that pumps protons to create a gradient, driving ATP synthase to make ATP.
I was asking you about ubiquinone and cytochrome C specifically, why we need them.
Ubiquinone and cytochrome C are mobile carriers in the electron transport chain that shuttle electrons between the big protein complexes, ensuring smooth energy transfer. Ubiquinone moves electrons from complexes 1 and 2 to 3, while cytochrome C carries them from complex 3 to oxidase. And we need them because without these connectors, the chain would break down, halting energy production for ATP. Want to dig into their specific chemical roles or move to something else?
Let's talk about the flow of electrons through the complexes. So explain to us what's going on, and kind of I want us to be more aware of the location too, like matrix, intermembrane space, outer leaflet.
All right, let's trace the electron flow through the electron transport chain ETC and nail down the locations. Electrons start in the mitochondrial matrix, where NADH donates them to complex 1, NADH dehydrogenase, located in the inner mitochondrial membrane. Complex 1 passes electrons to ubiquinone, a mobile carrier that floats in the inner membrane's lipid bilayer, including its outer leaflet. Ubiquinone shuttles electrons to complex 3, cytochrome BC1, also in the inner membrane, which then transfers them to cytochrome C, a protein moving along the inner membrane space side of the inner membrane. Cytochrome C delivers electrons to complex 4, cytochrome C oxidase, still in the inner membrane, which hands them to oxygen, forming water in the matrix. Complex 2, succinate dehydrogenase, also in the inner membrane, feeds electrons from FADH2 directly to ubiquinone, bypassing complex 1. As electrons move through complexes 1, 3, and 4, these complexes pump protons from the matrix to the inner membrane space, building a gradient that drives ATP synthase, located in the inner membrane, to produce ATP in the matrix. Want to focus on a specific complex's role or the proton movement?
I want to focus on the path of electrons entering. So where does it start? Where does it end?
Electrons enter the electron transport chain in the mitochondrial matrix, where NADH donates them to complex 1, or FADH2 donates them to complex 2, both in the inner mitochondrial membrane. The path ends at complex 4, also in the inner membrane, where electrons are transferred to oxygen, forming water in the matrix. Want to dive deeper into the starting molecules or the final step?
Yeah, let's talk and let's mention NADH, FMN, FE-S, and succinate and fumarate and all that shit.
All right, let's dive into the electron path and the electron transport chain, focusing on NADH, FMN, FAS, iron sulfur clusters, succinate, and fumarate. It starts in the mitochondrial matrix, where NADH, a high-energy molecule from the Krebs cycle, donates electrons to complex 1, NADH dehydrogenase. In complex 1, electrons first hit FMN, flavin mononucleotide, a prosthetic group that grabs them and passes them to a series of FAS clusters, which are iron-sulfur complexes that shuttle electrons within the protein due to their ability to cycle between oxidized and reduced states. From FAS clusters, electrons move to ubiquinone in the inner mitochondrial membrane. Meanwhile, complex 2, succinate dehydrogenase, takes succinate, also from the Krebs cycle, and oxidizes it to fumarate in the matrix, passing electrons directly to FAD, flavin adenine dinucleotide, which becomes FADH2, and then to ubiquinone, bypassing FMN and FAS. From ubiquinone, electrons flow through complex 3, cytochrome C, and complex 4 to oxygen, forming water in the matrix. These components, NADH, FMN, FAS, succinate, and fumarate, are key because they kickstart and sustain the electron flow for ATP production. Want to zoom in on any molecule's role or move to something like mitochondrial diseases?
Well, let's see how they relate with the synthesis of ATP a bit. And yeah, so tell me about the inner mitochondrial leaflet and why it's impermeable to charged species, but there's three systems in the membrane that they transport ADP and PI into the matrix, they synthesize ATP and they transport ATP into the cytosol. Explain to me this and why we strategically transfer these things.
All right, let's connect the electron transport chain, ETC, to ATP synthesis and the inner mitochondrial membrane's role. The inner mitochondrial membrane is nearly impermeable to charged species like ADP, phosphate, pi, and ATP due to its tightly packed lipid bilayer and limited ion channels, which maintains a proton gradient critical for ATP production. The ETC, as we discussed, uses electrons from NADH and FADH2 to pump protons from the matrix to the inner membrane space, creating this gradient. This proton motive force drives ATP synthase, a protein in the inner membrane, to synthesize ATP from ADP and pi in the matrix by allowing protons to flow back in, a process called oxidative phosphorylation. Now, three key transport systems manage the movement of ADP, pi, and ATP. First, the adenine nucleotide translocase, ANT, exchanges ADP from the cytosol for ATP from the
dude,
matrix.
This is great, but remember, I want you for the next 300 messages to answer to me with a maximum of like 3 sentences because I'm not a robot. Okay, having said that, let's talk a bit about adenine nucleotide translocase. And explain to us the concept of an antiporter, what it does, so why it transports ADP from the intramitochondrial membrane space into the matrix, and how exactly does the exchange with the ATP molecule occur. It's basically, I take my car to the mechanic in 2 pieces and the mechanic puts it in 1 piece, and then the mechanic brings it back to me, and the mechanic is basically, the cytosol, whereas,
Adenine nucleotide translocase, ANT, is an antipoder in the inner mitochondrial membrane that exchanges 1 ADP molecule from the inner membrane space near the cytosol for 1 ATP molecule from the matrix. An antipoder moves 2 different molecules in opposite directions, here using the electrochemical gradient to drive ADP in and ATP out, ensuring the matrix has ADP for ATP synthesis while supplying ATP to the cytosol for cell energy needs. The exchange is like a strict 1-for-1 trade, where ANT binds ADP, flips it into the matrix, and simultaneously binds and releases ATP to the cytosol, powered by the membrane's charge difference.
Interesting. But I don't understand. So adenine nucleotide translocase is the antipoder. But how did it get its name?
Adenine nucleotide translocase, ANT, is named for its role in translocating adenine nucleotides, specifically ADP and ATP, across the inner mitochondrial membrane. The term translocase reflects its function as a transport protein, an adenine nucleotide, specifically the molecules it handles, distinguishing it from other transporters. It's called an antipoder because it swaps 1 ADP for 1 ATP in opposite directions.
Okay, and let's talk about phosphate translocase now.
Phosphate translocase, also called the phosphate carrier, is a protein in the inner mitochondrial membrane that transports inorganic phosphate, pi, from the intermembrane space into the matrix, often as H2PO4-, in exchange for a hydroxyl ion, OH, or co-transporting with a proton, H+. This antiporter or symporter action ensures pi is available in the matrix for ATP synthase to combine with ADP to form ATP. Its function is critical for maintaining the phosphate supply needed for energy production in mitochondria.
So it's a second membrane transporter. It's helping with oxidative phosphorylation. How exactly, you have to tell me. And it helps with ATP synthesis, which you also have to tell me how it does it. So it transports phosphate and hydrogen ions into the matrix. I guess this helps with the ATP synthesis. This is a symporter though. Interesting. Where do we find the phosphate in the first place to go take it out?
Phosphate translocase, a symporter, transports inorganic phosphate, pi, and a proton, H+, from the inner membrane space into the mitochondrial matrix, using the proton gradient to drive uptake. This supplies pi for ATP synthase to combine with ADP during oxidative phosphorylation, directly supporting ATP synthesis in the matrix. Phosphate is sourced from the cytosol, where it's abundant from metabolic processes, and moves into the inner membrane space before being transported into the matrix.
What's ATP synthase and what do we mean by F-type ATPase?
ATP synthase is a protein complex in the inner mitochondrial membrane that synthesizes ATP from ADP and phosphate, pi, by using the proton gradient from the electron transport chain, acting like a tiny rotary motor as protons flow back into the matrix. It's called an F-type ATPase because it belongs to a family of enzymes, F-type, that can both synthesize ATP and, in some cases, hydrolyze it to pump protons, with F referring to its role in energy coupling. This process, part of oxidative phosphorylation, is crucial for cellular energy production.
Okay, and what's F0 and F1? So F0 is a proton channel and F1, who names this shit?
F0 and F1 are subunits of ATP synthase, where F0 is a proton channel embedded in the inner mitochondrial membrane, allowing protons to flow into the matrix, and F1 is the catalytic part in the matrix that uses this proton movement to synthesize ATP from ADP and phosphate. The names F0 and F1 come from historical biochemical studies, where F stands for fraction and the numbers indicate different components identified during enzyme purification. They're just labels stuck from lab work, not the most intuitive, but they describe the protein's two key functional parts.
Okay, sorry for the noise, guys. My housemate is eating red bell peppers. But yeah, let's talk a bit about the three different types of subunit regarding F0. So we have A, B, and C. And also we have 5 different types of subunit with F1.
The F0 subunit of ATP synthase have 3 main types of subunits, A, B, and C, where subunit A forms part of the proton channel, B anchors the complex to the membrane, and multiple C subunits create a rotating ring that drives proton movement. The F1 subunit has 5 types of subunits, alpha, beta, gamma, delta, and epsilon, with alpha and beta 3 copies each, forming a hexamer where beta catalyzes ATP synthesis, while gamma, delta, and epsilon form a central stalk that rotates to couple proton movement to ATP production. These subunits work together in the inner mitochondrial membrane to convert the proton gradient into ATP during oxidative phosphorylation.
And why is the gamma unit, what is going on with the gamma unit? It rotates. And what about the properties of the beta catalytic units?
The gamma subunit of ATP synthase's F1 portion forms a central stalk that rotates, driven by proton movement through F0's C ring, turning the F1 hexamer to trigger ATP synthesis. The beta subunits, 3 in F1, are catalytic, each with active sites that bind ADP and phosphate, changing shape as gamma rotates to synthesize ATP through conformational changes. This rotation and catalysis in the mitochondrial matrix convert the proton gradient's energy into ATP.
Interesting. Okay. So with the electrochemical energy generated, can you explain the difference in charge and the difference in proton concentration, and what even is electrochemical energy generated?
Electrochemical energy in the mitochondria comes from the proton gradient across the inner membrane, where a difference in proton concentration, more protons in the inner membrane space than the matrix, and charge, positive in the intermembrane space, negative in the matrix, creates a proton motive force. This gradient, built by the electron transport chain pumping protons out during electron transfer, has both a chemical component, higher H+ concentration outside, and an electrical component, charge difference across the membrane. ATP synthase harnesses this energy as protons flow back into the matrix, driving ATP synthesis from ADP and phosphate.
Okay. And what's the energy we yield from oxidation of one molecule of glucose then?
The oxidation of one glucose molecule through glycolysis, the Krebs cycle, and the electron transport chain, ETC, theoretically yields about 30-32 ATP molecules in eukaryotic cells, mostly via oxidative phosphorylation in mitochondria. This energy comes from NADH and FADH2, donating electrons to the ETC, driving proton pumping to power ATP synthase. The exact yield varies due to factors like proton leak or shuttle systems for NADH.
Explain this a bit more. Why 30 or 32? What's going on in specifically glycolysis for this?
Glycolysis in the cytoplasm breaks down one glucose into two pyruvate molecules, producing 2 ATP directly and 2 NADH, which can yield about 4-6 ATP via the electron transport chain, ETC, depending on shuttle efficiency. The Krebs cycle in the mitochondrial matrix per glucose generates 2 GTP, equivalent to ATP, 6 NADH, about 15-18 ATP via ETC, and 2 FADH2, about 3-4 ATP via ETC. The total of 30-32 ATP varies because NADH shuttling from glycolysis to mitochondria can be less efficient, and some energy is lost to proton leak across the inner membrane.
What's the difference between GTP and ATP?
GTP, granosine triphosphate, and ATP, adenosine triphosphate, are both nucleotide triphosphates that store and transfer energy. But GTP has a guanine base, while ATP has an adenine base. In cells, ATP is the primary energy currency, used widely for metabolic processes, while GTP is mainly used in specific pathways like protein synthesis and signaling, such as in the Krebs cycle, where GTP is produced but often converted to ATP. Their functional difference lies in their specific roles, with ATP being more universal and GTP more specialized.
Okay. What are uncoupling reagents?
Uncoupling reagents are chemicals that disrupt the proton gradient across the inner mitochondrial membrane, allowing protons to leak back into the matrix without passing through ATP synthase. This uncouples electron transport from ATP synthesis, reducing ATP production and dissipating energy as heat. Examples include 2,4-dinitrophenol, DNP, and ferrogenin in brown fat.
I don't get it. Tell me more about the uncoupling reagents. So normally we have electron flow and phosphorylation. In ADP, they are tightly coupled. So these uncouplers, they transport protons back in the matrix of the mitochondria and they bypass ATP synthase.
Uncoupling reagents break the link between electron flow and the electron transport chain, ETC, and ATP synthesis by allowing protons to move back into the mitochondrial matrix without going through ATP synthase. Normally, the ETC pumps protons into the inner membrane space, creating a gradient that drives ATP synthase to make ATP. Uncouplers, like 2,4-dinitrophenol, act like shuttles, carrying protons across the inner membrane, collapsing the gradient and wasting energy as heat instead of ATP. This bypass reduces ATP production, increases oxygen consumption, and can disrupt cellular energy balance.
Tell me more about DNP. Tell me about UCP1 too.
Two,4-dinoprofile, DNP, is an uncoupling reagent that shuttles protons across the inner mitochondrial membrane, collapsing the proton gradient and reducing ATP synthesis while increasing heat production. Historically used as a dangerous weight loss drug. Uncoupling protein 1, UCP1, or thermogenin, is a natural uncoupler in brown fat mitochondria identification, allowing proton leak to generate heat for thermoregulation, especially in newborns or hibernating animals. Both disrupt the link between electron transport and ATP synthesis, but DNP is toxic and unregulated, while UCP1 is a controlled physiological mechanism.
Okay, tell me about brown adipose tissue. Tell me about how it's specialized for heat generation. And talk to me about the high numbers of mitochondria that would contain thermogenin, UCP1.
Brown adipose tissue, BA, is specialized for heat generation, rich in mitochondria, packed with UCP1, thermogenin, which uncouples electron transport from ATP synthesis, leaking protons across the inner mitochondrial membrane to produce heat instead of ATP. Unlike white fat, which stores energy, BA's high mitochondrial density and UCP1 content enable rapid thermogenesis, crucial for maintaining body temperature in newborns, hibernating animals, or cold environments. Its brownish color comes from the mitochondria and abundant blood vessels, supporting its high metabolic activity for heat production.
Tell us a bit more about this.
Brown adipose tissue, BA, is loaded with mitochondria containing UCP1, thermogenin, which allows protons to leak back into the mitochondrial matrix, bypassing ATP synthesis and converting energy from electron transport into heat instead of ATP. This high mitochondrial density and UCP1 activity make BA highly effective for thermogenesis, helping regulate body temperature in cold conditions
But is this to the detriment of us producing ATP?
Yes. Brown adipose tissue's use of UCP1 for heat generation reduces ATP production because protons leak back into the mitochondrial matrix without driving ATP synthesis. This trade-off prioritizes thermogenesis over energy storage, which is critical for maintaining body temperature in specific conditions. However, BA is a small fraction of body tissue, so its impact on overall ATP production is limited.
So you said that the uncouplers separate the ATP synthase stuff from the protons, right?
Yeah. Uncouplers like DNP or UCP1 let protons leak back into the mitochondrial matrix without passing through ATP synthase, breaking the link between the proton gradient and ATP production. This means the energy from the electron transport chain goes to heat instead of making ATP. It's like bypassing the machine that turns energy into useful fuel.
I see. So I wonder in one sentence, if someone had to take one main takeaway from this video, what would it be?
The mitochondrial respiratory chain generates ATP by transferring electrons through protein complexes, but uncouplers like UCP1 and brown fat prioritize heat production over ATP synthesis for thermoregulation. Want to wrap up with anything else?
That's perfect. So guys, thank you for watching part 1. I'm trying to keep these videos 30 minutes long. I'm going to do part 2. Make sure to watch part 2 to see the rest of the topics we said we would discuss. Thank you. Peace.